Once again, welcome to the course on basic genetics and our colleagues Dr. Richa Are, Professor Madhu Gavaldas Tapadia and myself Jagat Kumar Roy, we will be taking these lectures. This is week 10, lecture 2 and the title is G banding. G banding is one of the important technique of earlier days when the chromosome biology was in its infant. This picture, the picture you are seeing, this is human chromosome karyotype. This has already been described to you in my last lecture. What kind of problems we face in making these karyotypes? Identifying the chromosome correctly and pairing them often becomes difficult. For example, I am showing this is a chromosome. Whether this pairs best with this or this pair best with this sometimes becomes a confusing condition. Same way here in group C, so many different chromosomes are there. Whether this and this pairing is correct or there is some other option that is correct, that means one is not very sure. So that is why if we have some kind of a situation where these chromosomes are having a banding or having a permanent kind of impression which is not changed from time to time, that would have been the best thing. One tried many different banding techniques and these chromosome bandings provided easy identification of each chromosome accurately. Out of many bandings, those got initiated during those times, G banding is the most common, most reliable one since those days, even today. In G banding, what one does, metaphase chromosome preparation, as we described in the previous lecture, they are made. The chromosome preparation is made on slide. The slide is allowed to age for 2 to 3 days. Why aging? Because if the chromosome is not so much dry, when further treatment is given, the chromosomes may lose morphology, but with aging, the morphology remains better. So metaphase chromosome preparation is done. Then 1 ml of trypsin stock solution. Trypsin is a protease and this is 30 milligram per ml concentration. This trypsin is made in 0.9% sodium chloride solution. So this 1 ml is diluted to 50 ml. That means now you have a coupling jar full of the trypsin solution. And then adjusted the pH to 7.5 to 7.8, checking with a pH paper. Usually 1 to 2 drops of 1 molar NaOH is sufficient for making this pH. This pH is not exactly that one has to have 7.5 to 7.8, that much variation is fine. And then, so you have one coupling jar with trypsin solution, 50 ml. There has to be another coupling jar where a buffer should be there, that is phosphate buffer. This phosphate buffer is nothing but KH2PO4 solution and Na2HPO4 solution made in such a way that the final pH is 7.5. So that is how it is said solution 1, 9.073 gram of KH2PO4 in 1 liter. The other one 11.87 gram of Na2HPO4. So this is Na2. This is basic, this is KH2, this is slightly acidic, and so this is also in 1 liter of distilled water. You mix the two in a equal ratio so that finally the solution is a buffer. This is a phosphate buffer. The third coupling jar has to be with Giemsa stain. 7 to 10%, 5 to 10% is also fine of Giemsa stain in phosphate buffer is made ready. So first, dip the slide in trypsin for just a few seconds, let us say 5 seconds. This time has to adjusted or this time has to get once standardized. So for 5 seconds, then immediately rinse the slide in phosphate buffer after trypsin exposure and then stain the slide for 5 to 10 minutes with Giemsa. Observe this slide under the microscope. If good banding is there, then it is fine. Otherwise, if the chromosome morphology has gone bad, reduce the time. If chromosome morphology is fine and banding is not so well, you can increase the time. So this is how the banding is done. After the banding, the G-banded chromosomes are seen like this. So this is the picture of G-band. You can see here each chromosome, if you see. These chromosomes are banded. These chromosomes are banded. They have dark bands. So these dark bands are called as G positive band, and the light band, this is the light part, this is known as G negative band. So the entire chromosome, it is like this, that if suppose you have a wall, you have different rooms made, the top is all cemented, but one may have brick joined with mud, another may have brick joined with lime and other things which were used in earlier days, and the third is with cement. If you want to test what kind of wall is this, one has to remove the cement layer. Same thing in chromosome. When we treated with trypsin, we removed some of the superficial proteins from chromosome. The chromosome as such architecture wise, it has at some places it has the more thickening or it has tight coiling of the DNA. At some places it has a thinning or less tight packing of the DNA. So that is how the chromosome architecture is having less thick, more thick, less thick, more thick, like alternative, like this. So by removing the outer protein layer or superficial layer, one is now seeing the chromosome architecture. And this is permanent. It is not that this will change with time or change with different developmental stage. So this is the G-banded human. Now, with this, if this is the plate, and if we again do the same thing that you cut out all the chromosomes, the same way you are cutting out with the scissors, and then if you arrange them on the paper pairwise, you are able to make the karyotype. The same karyotype is being shown here. Here you can see that now here there will not be any mistake. You yourself will be able to see that these bands are very constant. These bands, what is present in one chromosome, the same bands are present in another chromosome. You can see the thickness, you can see the bands, everything is remaining same. So this way you will not be able to make any mistake, and chromosomes are paired. The chromosome karyotyping is done in the same way that chromosome 1, 2 and 3, the metacentric ones, they are placed in group A. Chromosome 4 and 5, they are placed in group B. Chromosome 6 to 12, they are placed in group C. This is the X chromosome is also part of this. Here this has been separated, that is why it is not here. Then group D is having 13 to 15 chromosome. This is the acrocentric chromosome with satellite. You can see the satellite very clearly here. This is the satellite. This is the satellite. And then chromosome 16 to 18, these are small submetacentrics. Then 19, 20, small metacentric. Then 21, 22, very small acrocentric with satellite. So here this is the satellite. This is the satellite. This is the satellite. This is the satellite. And if Y chromosome is there, the shape is the same, but that will not have satellite. So this is your G banding. And now let us see one chromosome or one chromosome pair. You can see these bands. These are the bands which are same in both the chromosomes. So the G band positive regions, what you call it as G band positive regions, that means dark band with Giemsa. So these are G band positive regions. And these regions are heterochromatic and they are AT rich. AT rich means in the chromosome you have DNA. Chromosome is made up of DNA that we had described in earlier week. So these DNA, whatever DNA we have, so that DNA is having the nitrogenous bases, the sugar and phosphate. The nitrogenous bases, they are adenine, guanine, cytosine and thymine. So adenine in the two strand, this is two opposite strand, adenine pairs with thymine and guanine pairs with cytosine. So that is what you call it as A with T and G with C. So if suppose in a region you have more A in one, T in another, so that will be AT rich. So that is what I said, that these regions are heterochromatic and they are AT rich. Usually most of the heterochromatin, except in some organism, they are AT rich. So these are heterochromatic regions. The G band negative, this is G band negative region. These are Giemsa light staining region and they are euchromatic. Euchromatin means true chromatin, heterochromatin means different chromatin or condensed chromatin. What are the difference between euchromatin and heterochromatin? Even during replication of the DNA, euchromatin replicates earlier, heterochromatin replicates late. Euchromatin has most of the functional genes, heterochromatin has less or no functional gene. So the G band negative regions are light stained regions, euchromatic and non-AT rich. Do not call it as GC rich, because non-AT means more A and T is not there. It may have distribution of AT and GC. So with this, I come to the activity and discussion. Once again, I'll say, take a printout of human G-banded metaphase plate and prepare karyotype by yourself. Once you practice, if you do karyotype once, twice or thrice, you yourself will be confident. Why do you treat chromosomes with trypsin and not with any other proteases? One can try with other proteases. But once a technique is standardized, one tends to do the same thing. And that is how with trypsin this has been standardized, and that is why one does not want to change. And now this has become a very standard kind of banding even for today. So everybody follows G banding, and these G banded, as we see these, we will go for these banding nomenclature. So they are constant, they will remain constant in a chromosome, and they have been numbered. They have been numbered with reference to the centromere. If this is the centromere, they have been numbered as 1, 2, 3, 4, 1, 2, 3, 4, like this. So the question is, are G-band positive and G-band negative regions always remain constant? I will say yes, they will remain constant. So these things are to be discussed thoroughly by you, and that will be the best exercise. Thank you.